Hello and welcome everyone. Myself Mijanur Rahaman, and today my topic of discussion is hemoglobin and iron metabolism. So today we will discuss about the introduction part, then normal hemoglobin content, function, structure, then types of normal hemoglobin, then abnormal hemoglobin derivatives, then synthesis and then destruction and then iron metabolism. So today our topic is very big. So let us start. Let us introduce first. Hemoglobin is the iron containing coloring matter of the red blood cell, and it is chromoprotein forming 95% of dry weight of RBC and 30% to 34% of weight. And function of hemoglobin is to carry the respiratory gases, oxygen and carbon dioxide. And it also acts as buffer, and its molecular weight is around 68,000 Dalton. Now normal hemoglobin content. Average hemoglobin content in the blood is about 14 to 16 gram per dl, and the values of varies depending upon the age and sex of the individual. At the time of birth, it is present 25 gram per dl. Third month, 20 gram per dl. After 1 year, 17 gram per dl, and from puberty and onwards, 14 to 16 gram per dl. At the time of birth, hemoglobin content is very high as because is the increased number of RBC. Then sex, in case of adult male it is 15 gram per dl, and in adult female it is 14.5 gram per dl, and it may ranges also. In case of male it is around normal is 12 to 16 gram per dl, and in case of female it is 10 or 11 to 16 gram per dl is normal. Then function of hemoglobin. Transport of respiratory gases. Main function of hemoglobin is to transport the respiratory gases like oxygen from the lungs to tissues and carbon dioxide from the tissues to lungs. Then transport of oxygen. When oxygen binds with hemoglobin, a physical process called oxygenation occurs and resulting in the formation of oxyhemoglobin. And the iron remains in ferrous state in this compound, and oxyhemoglobin is an unstable compound and the combination is reversible. So when more oxygen is available, it combines with the hemoglobin, and whenever oxygen is required, the hemoglobin can release oxygen readily. And when oxygen is released from oxyhemoglobin, it is called reduced hemoglobin or ferrohemoglobin. Then transport of carbon dioxide. Whenever the carbon dioxide binds with the hemoglobin, it is known as carb hemoglobin, and it is also an unstable compound, and the combination is reversible when the carbon dioxide can be released from this compound. The affinity of hemoglobin for carbon dioxide is 20 times more than that of oxygen. Then buffer action. Hemoglobin acts as buffer and plays an important role in acid-base balance. Now, function is complete. Now we'll start the structure of hemoglobin. Hemoglobin is a conjugated protein. Okay, it consists of a protein combined with an iron-containing pigment, and the protein part is globin, and the iron-containing pigment is heme. Heme also forms a part of the structure of the myoglobin, oxygen binding pigment in muscle, and neuroglobin, oxygen binding pigment in brain. Then iron. Normally it is present in ferrous or Fe2 plus form, and it is in unstable or loose form, and in some abnormal condition the iron is converted into ferric or Fe3 plus state, which is a stable form. Then porphyrin. The pigment part of heme is called porphyrin, and it is formed by 4 pyrrole rings or tetrapyrrole called 1, 2, 3 and 4. The pyrrole rings are attached to one another by methane or CH4 bridges, and the iron is attached to N of each pyrrole ring and N of globin molecule. Then globin. Globin contains 4 polypeptide chains. Among the 4 polypeptide chains, 2 are chains and 2 are alpha chains and 2 are beta chains. And the alpha chain molecular weight is 15,126 Dalton. It has 141 amino acid, and beta chain is slightly heavier, which is 15,866 Dalton, and its amino acid is 146. Now types of normal hemoglobin. Hemoglobin is of two types. One is adult hemoglobin or HbA, and one is fetal hemoglobin HbF. Another hemoglobin is also present like HbA1, A2. Now, replacement of fetal hemoglobin by adult hemoglobin starts immediately after birth. It is completed about in the 10 month to 12 month after birth. Both the types of hemoglobin differ from each other structurally and functionally. Now, structural difference. In adult hemoglobin, the globin contains two alpha chains and two beta chains, and in fetal hemoglobin, there are two alpha chain and two gamma chain instead of beta chains. Now, functional difference. Functionally, fetal hemoglobin has more affinity for oxygen and than that of the adult hemoglobin, and the oxygen hemoglobin dissociation curve of the fetal blood is shifted to left. Now, we will discuss about the abnormal hemoglobin. Abnormal types of hemoglobin or hemoglobin variants are the pathologic mutant form of hemoglobin, and these variants are produced because of structural changes in the polypeptide chains caused by mutation in the genes of the globin chains, and most of the mutations do not produce any serious problem. Occasionally, few mutations result in some disorder. There are two categories of abnormal hemoglobin. One is hemoglobinopathies and one is hemoglobin in thalassemia and related disorder. Hemoglobinopathy is a genetic disorder caused by abnormal polypeptide chains of hemoglobin. Some of the hemoglobinopathies are hemoglobin S. It is found in sickle cell anemia, and in this the alpha chains are normal and beta chains are abnormal. Hemoglobin C, the beta chains are abnormal and it is found in people with hemoglobin C disease, which are characterized by mild hemolytic anemia and splenomegaly. Then hemoglobin E. Here also beta chains are abnormal and it is present in the people with hemoglobin E disease, which is also characterized by mild hemolytic anemia and splenomegaly. Then hemoglobin M. It is the abnormal hemoglobin present in the form of methemoglobin, and it occurs due to mutation of genes of both in alpha and beta genes, and which results in abnormal replacement of amino acid, and it is present in babies affected by hemoglobin M disease or blue baby syndrome, and it is an inherited disease characterized by methemoglobinemia. Now, hemoglobin in thalassemia and related disorder. In thalassemia, different types of abnormal hemoglobins are present. The polypeptide chains are decreased, absent, or abnormal. In alpha thalassemia, the alpha chains are decreased, absent, or abnormal, and in beta thalassemia, the beta chains are decreased, absent, or abnormal. And some of the abnormal hemoglobin are found in thalassemia are hemoglobin G, H, I, Bart's, Kenya, Leper, and Constant Spring. Now, abnormal hemoglobin derivatives. Hemoglobin derivatives refer to a blood test to detect and measure the percentage of abnormal hemoglobin derivatives. Hemoglobin is the only carrier for transport of oxygen without which tissue death occurs within few minutes. When hemoglobin is altered, its oxygen carrying capacity is decreased and resulting in lack of oxygen. So, it is important to know about the causes and the effects of abnormal hemoglobin derivatives. Now, abnormal hemoglobin derivatives are found by carbon monoxide poisoning or due to some drugs like nitrites, nitrates, and sulfonamides. Now, abnormal hemoglobin derivatives are carboxyhemoglobin, methemoglobin, and sulfahemoglobin. Now, normal percentage of the hemoglobin derivatives in total hemoglobin. Carboxyhemoglobin is 3 to 5%, methemoglobin is less than 3%, and sulfahemoglobin is trace or undetectable amount. Abnormally high levels of hemoglobin derivatives in blood produce serious effects. These derivatives present in transport of oxygen resulting in oxygen lack in tissues which may be fatal. Let us start about the carboxyhemoglobin. Carboxyhemoglobin or carbon monoxyhemoglobin is the abnormal hemoglobin derivative formed by the combination of carbon monoxide with hemoglobin. Carbon monoxide is a colorless and odorless gas. Since hemoglobin has 200 times more affinity for carbon monoxide than oxygen, it hinders the transport of oxygen resulting in tissue hypoxia. Normally 1 to 3% of hemoglobin is in the form of carboxyhemoglobin. Now sources of carbon monoxide: charcoal burning, coal mine, deep wells, then underground drainage system, exhaust of gasoline engines, then gases from guns and other weapons, heating system with poor or improper ventilation, smoke from fire, and tobacco smoking. Then sign and symptoms of carbon monoxide poisoning: while breathing air with less than 1% of carbon monoxide, the hemoglobin saturation is 15 to 20%, and mild symptoms like headache and nausea appear. Then, while breathing the air with more than 1% carbon monoxide, the hemoglobin saturation is 30% to 40%, and it causes serious symptoms like convulsion, cardiorespiratory arrest, and unconsciousness and coma. When hemoglobin saturation increases above 50%, death occurs. Then methemoglobin. Methemoglobin is the abnormal hemoglobin derivative formed when iron molecule of hemoglobin is oxidized from normal ferrous state of or to ferric state. Methemoglobin is also called ferrihemoglobin. Normal methemoglobin level is 0.6% to 2.5% of total hemoglobin. Under normal circumstances, also body faces the threat of continuous production of methemoglobin, but it is counteracted by the erythrocyte protective system called nicotinamide adenine dinucleotide or NADH system, which operates through the two enzymes. One is diaphorase 1 nicotinamide adenine dinucleotide phosphate NADPase dependent reductase. It is responsible for 95% of the action, and diaphorase 2, that is NADPase dependent methemoglobin reductase, responsible for 5% of the action. These two enzymes prevent the oxidation of ferrous iron into ferric iron. Then methemoglobinemia. Methemoglobinemia is a disorder characterized by high level of methemoglobin in blood, and it leads to tissue hypoxia which causes cyanosis and other symptoms. Now, causes of methemoglobinemia. Methemoglobinemia is caused by a variety of factors. Common factors in daily life like well water contaminated with nitrates and nitrites, fires, laundry ink, matchsticks and explosives, meat preservatives when contain nitrates and nitrites, mothballs, naphthalene balls, room deodorizer, polypeptides, propellants. Then exposure to industrial chemicals such as aromatic amines, fluorides, irritant gases like nitrous oxide and nitrobenzene. Then propylene glycol denitrate. Then drugs, antibacterial drugs like sulfonamides. Then antimalarial drugs like chloroquine. Then antiseptics. Then inhalant in cyanide antidote kit. Then local anesthetics like benzocaine. Now, hereditary trait. Due to deficiency of NADH dependent reductase or presence of abnormal hemoglobin M, the hemoglobin M is the common in babies affected by blue baby syndrome, a pathological condition in infants and characterized by bluish skin discoloration or cyanosis and caused by congenital heart defect. Then, sulfhemoglobin. Sulfhemoglobin is the abnormal hemoglobin derivative formed by the combination of hemoglobin with hydrogen sulfide, and it is caused by drugs such as phenacetin or sulfonamides. Normal sulfhemoglobin level is less than 1% of the total hemoglobin, and sulfhemoglobin cannot be converted back into hemoglobin. Only way to get rid of this form of body is to wait until affected RBC with sulfhemoglobin are destroyed after their lifespan. Now, blood level of sulfhemoglobin. Normally, very negligible amount of sulfhemoglobin is present in blood, which is non-detectable. But when its level rises above 10 gram per dl, cyanosis occur. Usually, serious toxic effects are not noticed. Now, synthesis of hemoglobin. Synthesis of hemoglobin actually starts in pro-erythroblastic cells. However, hemoglobin appears in the intermediate normoblastic stage only, and production of hemoglobin is continued until the stage of reticulocyte. Heme portion of hemoglobin is synthesized in the mitochondria, and the protein part or globin is synthesized in the ribosome. Now, synthesis of heme. Heme is synthesized from succinyl-CoA and glycine. The sequence of events are in synthesis of hemoglobin. First step in heme synthesis takes place in the mitochondria. Two molecules of succinyl-CoA combines with the two molecules of glycine and condense to form delta amino levulinic acid or ALA by ALA synthase. Now ALA is transported to the cytoplasm. Two molecules of ALA combine to form porphobilinogen in the presence of ALA dehydratase. Now porphobilinogen is converted into uroporphyrin by uroporphobilinogen 1 by uroporphobilinogen 1 synthase. Now uroporphobilinogen 1 is converted into uroporphobilinogen 3 by porphobilinogen 3 convertase co-synthase. Now from uroporphobilinogen 3 is a ring structure called coporphofibrinogen 3 is found by uroporphobilinogen decarboxylase, and coproporphobilinogen 3 is transported back to the mitochondria where it is oxidized to form protoporphyrinogen 9 by coporphyrinogen. So what happens here? Coproporphyrinogen 3 is transported back to the mitochondria and where it is oxidized to form protoporphyrinogen 9 by coproporphyrinogen oxidase, and protoporphyrinogen 9 is converted into protoporphyrin 9 by protoporphyrinogen oxidase, and protoporphyrin 9 combines with iron to form heme in the presence of ferrochelatase. Now formation of globin. Polypeptide chains of globin are produced in the ribosomes. There are 4 types of polypeptide chains, namely alpha, beta, gamma and delta chains, and each of these chains differs from others by the amino acid sequence. Each globin molecule is formed by the combination of 2 pairs of chains, and each chain is made up of 141 to 146 amino acid. And adult hemoglobin contains 2 alpha chains and 2 beta chains, and fetal hemoglobin contains 2 alpha chains and 2 gamma chains. Now configuration. Each polypeptide chain combines with one heme molecule. Thus, after the complete configuration, each hemoglobin molecule contains 4 polypeptide chains and 4 heme molecule. Now, if we see the figure of synthesis of hemoglobin, then in the mitochondria, the succinyl-CoA plus glycine along with ALA synthase, it is converted into delta amino levulinic acid, which comes into the cytoplasm and converted into the 2 molecules of delta amino levulinic acid, and next step, with the help of ALA dehydratase, it is converted into proporphobilinogen, and it is synthased to uroporphobilinogen 1, which is co-synthase uroporphobilinogen 3, then decarboxylase by coproporphyrinogen 3, and it is come again into the mitochondria coproporphyrinogen 3 with oxidase to form protoporphyrinogen 9, with further oxidase to form protoporphyrin 9, which is along with ferrochelatase, it is converted into the heme, and heme comes out of the mitochondria, and globin is also attached with it, and formation of hemoglobin is done. Now substances necessary for hemoglobin synthesis. Various materials are essential for the formation of hemoglobin in the RBC. Now destruction of hemoglobin. After the lifespan of 120 days, the RBC is destroyed in the reticuloendothelial system and particularly in the spleen. and the hemoglobin is released into the plasma. Soon the hemoglobin is degraded in reticuloendothelial cells and split into globin and heme. And globin is utilized for resynthesis of hemoglobin, and heme is degraded into iron and porphyrin. Iron is stored in the body by as ferritin and hemosiderin, and which are radiolized for the synthesis of new hemoglobin. And porphyrin is converted into a green pigment called biliverdin. And in human being, most of the biliverdin is converted into yellow pigment called bilirubin, and bilirubin and biliverdin are together called bile pigments. Now, iron metabolism. Importance of iron. Iron is an essential mineral and an important component of proteins involved in oxygen transport. So, human body needs iron for oxygen transport. Iron is important for the formation of hemoglobin and myoglobin, and iron is necessary for the formation of other substances like cytochrome oxidase, peroxidase, and catalase. Now we will start the normal value and distribution of iron in the body. Total quantity of iron in the body is about 4 gram. And approximate distribution of the iron in the body is as follows. In the hemoglobin it is 65 to 68%, and in the muscle as myoglobin it is 4%, and as intracellular oxidative heme compound it is 1%, and then in the plasma as transferrin it is 0.1% and stored in the reticuloendothelial system as 25 to 30%. Then dietary iron. Dietary iron is available from two forms called heme and non-heme. Heme iron is present in fish, meat, and chicken. Iron in those sources is found in the form of heme, and heme iron is absorbed easily from intestine. And non-heme iron in the form of non-heme is available in vegetables and grains and cereals, and non-heme iron is not absorbed easily as heme iron. Then the cereals, flours, and products of grains which are enriched or fortified or strengthened with iron become good dietary sources for non-heme iron, particularly for children and women. Then absorption of iron. Iron is absorbed mainly from the small intestine, and it is absorbed through the intestinal cells, enterocytes, by pinocytosis and transported into the blood. Now, bile is essential for the absorption of iron, and iron is present mostly in ferric epithelial sperm, and it is converted into ferrous sperm or Fe2+, which is absorbed into the blood, and hydrochloric acid from gastric juice makes the ferrous iron soluble so that it could be converted into ferric iron by the enzyme ferric reductase and form enterocytes. From enterocytes, the ferric iron is transported into blood by a protein called ferroprotein, and in the blood the ferric iron is converted into ferrous iron and transported again. Now, transport of iron. Immediately after the absorption of into the blood, iron combines with a beta globulin called apotransferrin secreted by liver through bile, resulting in the formation of transferrin, and iron is transported into the blood in the form of transferrin. Iron combines loosely with globin and can be released easily at any region of the body. Now, storage of iron. Iron is stored in the large quantities in reticuloendothelial cells and liver hepatocytes. In other cells also it is stored in small quantities, and in the cytoplasm of cells iron is stored as ferritin in large amount. Small quantity of iron is also stored as hemosiderin. Daily loss of iron. In males about 1 milligram of iron is excreted every day through feces, and in females the amount of iron loss is very much high. This is because of menstruation, and 1 gram of hemoglobin contains 3.34 mg of iron. Normally 100 ml of blood contains 15 gram of hemoglobin and about 50 milligram of iron. So 3.34 into 15, it is 50 milligram. So if 100 ml of blood is lost from the body, there is a loss of about 50 milligram of iron. So in females, during every menstrual cycle, about 50 ml of blood is lost, by which 20 milligram of iron is lost. This is why the iron content is always less in females than in males. Then iron is lost during hemorrhage and blood donation. Also, if 450 ml of blood is donated, then about 225 milligram of iron is also lost. Now, regulation of total iron in the body, absorption and excretion of iron are maintained almost equally under normal physiological condition. When the iron storage is saturated in the body, it automatically reduces the further absorption of iron from the gastrointestinal tract by feedback mechanism. Factors which reduce the absorption of iron: stoppage of apotransferrin formation in the liver so that the iron cannot be absorbed from the intestine, and reduction in the release of iron from transferrin. So the transferrin is completely saturated with iron and further absorption is prevented. So this is for now. Today we have completed a major topic. So if you have any query or doubt, please comment down in the comment section, and thank you so much for watching. And do like, share and subscribe to my channel for supporting me and my channel, and thank you so much.